In DNA replication, we're going to have four main proteins, which is going to be helicase, DNA polymerase 1, primase, and ligase, as well as SSB proteins and gyrase are going to make their appearances. So as helicase runs down the DNA protein, it's going to unzip it, and gyrase is going to be used to relieve the supercoiling that occurs. Here we'll see the full DNA protein as it is unzipped, and SSB proteins are used to keep the DNA strand from coiling back on itself. Here, primase is going to come in, and it's going to lay down some primers, which are actually made of RNA, and we'll see that our 3' to 5' is going to be our template strand, while our 5' to 3' is going to be our coding strand. And so here DNA polymerase comes in after it is indicated by the primers to come in and start replicating. And once it has replicated, you'll see that the leading strand is going in the direction of the helicase, while the lagging strand is going in the opposite direction. And +1 is to indicate the origin of where replication occurs. Sorry, my cats are fighting. So for the lagging strand, we'll see that DNA polymerase is actually going to have to detach from replicating, and a new primer will have to be set because it's going in the opposite direction of helicase. And so again, it just continues to do that, and eventually we'll have these fragments where we have the primers and then the fragments, which are going to be called Okazaki fragments. And so DNA polymerase can actually move in the 5' direction, 5' to 3' direction, doing this thing called exonuclease, where it pretty much proofreads the DNA, and it takes out the primers, so then ligase can come in and work with the Okazaki fragments here and glue them back together to make a whole DNA strand. And so the question is, if 5' to 3' exonuclease didn't happen, what would occur? And pretty much, again, the DNA polymerase 1 would not be able to perform its function of proofreading the DNA as well as removing the primers so that the Okazaki fragments could be repaired properly. Thank you.